Okay, so Mendel lived in the 19th century. He was a monk, and he was called the father of genetics because he discovered how genes were passed on from generation to generation. Now, he did his experiments on pea plants, but I'm going to talk about experiments done in a fruit fly, little fruit fly about 3 millimeters long, called Drosophila. Now, Drosophila, an awful lot of genetic experiments we carried out in Drosophila because its life cycle means that it can produce a load of eggs every week, and they will hatch out and form mature flies within a week. So the experiments can be done quite quickly. You cross two flies, they'll produce hundreds of eggs, so you get a large sample size. And also the genetics of Drosophila is pretty similar to that of human beings. It's only got 4 chromosomes, but it does have an X and Y chromosome. Plants, a lot of plants are a little bit strange. They have multiple copies of genes and polyploidy and things. So this is a good model to use. Now, the gene we're going to look at here, we'll call it E. That is normal eyes, normal eye color, which is ready brown for these flies. And E is a mutated gene, and that will give the fly white eyes. E is recessive, and so, homozygous dominant, they'll have normal eyes. Heterozygous will have normal eyes. Homozygous recessive, they will have white eyes. Now, another thing is Mendel didn't know anything about chromosomes or genes. He called them factors. And so we're going to do this with the benefit of hindsight, if you like. We're going to know where the genes are sitting on the chromosomes. We know what happens during meiosis. You halve the number of chromosomes, and that we have a pair of homologous chromosomes. So we're going to talk about this from our perspective rather than just from Mendel's perspective. So what we've got here is we have got an individual fly. I've only drawn one of it's got 4 pairs of chromosomes. I've only drawn one pair, one homologous pair. The red would be from its father, red from the mother, whatever. And the gene for eye color is on one of those chromosomes. So, and there is the other individual. Now they're going to mate. Here, and this line, these are the gametes that they can produce, the sex cells. So, of course, during meiosis, the homologous pairs are separated and the chromatids are separated. Most importantly, it goes from having the diploid number of chromosomes to the haploid number. Then, during fertilization, well, the following can occur, can't it? You could have these two coming together, in which case you'd have an individual with that genotype, or if you got those two, you would have it as that, or if you got those two, and finally you could have that one. So let's look at the phenotypes of those. So hopefully we should know that the phenotypes right here, these are all going to be it's called wild type eye color. So normal eyes, or wild type normal eyes, and this one will have white eyes. So if a Drosophila, you've crossed 2 little Drosophila, the female will lay over 100 eggs, and then you can count the offspring and see their white eyes. Do this experiment, and you should get a 3-to-1 ratio of normal eyes to white eyes. Okay, so that none of that is much different than we've done at GCSE. Normally when you do these things, you do them in a table like this rather than showing where the chromosomes go. You just draw, like the gametes would go up there. They're the female possible gametes. These are the male gametes, and then you'd put, you do this kind of business here, which you've all done before. Use one of those squares. Right, now this is what happens when we've just looked at one trait being examined here, and that trait was eye color. At A level, you need to be able to know what happens when you're looking at 2 traits. So that would be called, this would be called a monohybrid cross, one trait. But we need to think next about a dihybrid cross. what would happen there? Well, so here I've got I've drawn some. Let's rub this out here. Start off with. Right. What we've got is we have got one individual. That is the male, if you like, and we're going to cross that with this female. Now, I've got two traits here. I've got the eye color one, which I've done in green, and we've got another recessive trait, which I've done in blue. And that sits on a different chromosome, and that's really important. So it's on a different chromosome to the gene carrying the eye color. We've got B stands for, that's, you've got normal body color, which is kind of a pale brown for Drosophila. The mutated gene gives the fly a black body color. And it's the black body color is recessive, so you need a double dose of the mutated gene in order to get the black body color. Okay, so here are the two individuals. And here are, right, I'm just going to draw it from, because it's the same for this one, but I'm just going to draw, these are the gametes that it can produce. These are the possible gametes. So if you look at the genotypes of those gametes, this one would be that, this one would be that, and the final one would have a mutated gene both for eye color and for body color. And you would get equal numbers of all of those mutated genes, or equal numbers of all of those gametes produced by one fly. Right, now why is that? Well, the reason why is, if you remember in meiosis, I draw a quick cell here, and I've only drawn obviously 2 chromosomes of the Drosophila. It's got 4, as I said before, but at meiosis, the homologous pairs are going to line up pretty close to the cell. So let's say that the homologous pairs are going to line up like that, and the spindles are going to form, and it's going to separate those homologous chromosomes in meiosis first division. So in that case, you're going to get a gamete like that with the black, right? And this gamete would have the longer chromosome red and the shorter chromosome red. But we all know that you get independent assortment of chromosomes in meiosis, and equally likely what could happen in the meiotic division is you could have the chromosomes lined up that way, in which case you produce different gametes. The gametes then would be, you get the black one and the red one with a short red one, and this one you get the long red one and the short black one. So you get different gametes. And the way the chromosomes do that is just purely random, and so all possibilities are equally likely. Okay, so now it's going to be a bit confusing to show what happens when these gametes meet up with other gametes, like I did before by drawing the lines. So I'm going to draw the square thing which you've used before at GCSE. Okay, so if you see here, these are the 4 possible gametes that the female can produce. Doesn't matter if it's male or female on the top. And here are the gametes that the male can produce here. So let's just take that one and that one, for example. So the phenotype there, the genotype, sorry, would be small e, big B, big B. So that individual would have white eyes, but it would have a normal body color. Now let's see what happens for the whole thing there. Okay, so what I've written down here are all the possible phenotypes that you can have. There are 4 possible phenotypes. You can have normal eyes and normal body color. To color code them. You could have white eyes and a normal body color. You could have normal eyes and the black body color, or you could have the mutated version of both genes, so white eyes and black body color. Right, so let's count them up and see the possibilities. So these red ones then, so this is normal eye, normal body, would be that one. Yeah. and that one. So we should count them up. That is 9 of those. Okay, let's see the ones with white eyes, but a normal body. So we need 2 little e's for white eyes. So yeah, that would be 1, and that would be 1, and that would be 1. Okay, so there's 3 of them. What would be normal eyes and black body? So you need 2 little b's for this time. So that would be one of these types. So with that, and so with that, so we get 3 of those. And finally, we just get 1 with the double mutation there. Okay, so we get 1 of them. So the ratio we would get in the dihybrid cross, then you get a 9 to 3 to 3 to 1 ratio. So if you did this with 2 Drosophila and they mated and you got like 250 offspring off them, then well, 9, that all adds up to 16, does it? So 9/16 of 250, whatever that be, would be, you'd expect to be normal. 3/16 of 250 would have white eyes but a normal body. 3/16 normal eyes but a black body, and then 1 in 16 you would expect with the double mutation. That would be the ratio that you'd expect there. 9 to 3 to 3 to 1. Okay. So that's fine, that's a dihybrid cross, but there was a big assumption there, and that was we assumed, and Mendel saw this 9 to 3 to 3 to 1 ratio, we assumed that both of those genes were on separate chromosomes. That is the case with those 2 particular mutations in Drosophila. They are on separate chromosomes. Let's see what would happen if they were on the same, let's pretend they were on the same chromosome, right? Because, okay, so here we have got the 2 individuals. This is the male and the female, if you like. Okay, and we're going to cross those 2 together. Let's have a look at the gametes that they can produce. Right, so that would be like this. That would be like that. And that would be like that. And okay, so you may have noticed then there's no possibility of for example, that one. And there's no possibility of having that one. It's impossible because the genes are on the same chromosome. So let's do what we would expect for that. So I'm going to draw a diagram here. So the gametes you can have here are, that's the female gametes. Yeah, and on the vertical we'll have the male gametes. Let's put in the genotypes of the offspring then. So that one would be like that. And so let's have a look at those. Well, this would be, I think we color coded it in red before. It's sort of a bit out of reach up there, so I won't draw it, but we colored that in red. So that is going to be the normal body, sorry, normal eye, normal body. Okay, now this one is also, that's going to be the same. And that one's going to be the same. And the last one, well, this would be the other one. That would be the double mutation. So that would be white eye and black body. And if you look, we didn't get any of the other. It's impossible to get the other one. So if I think the yellow one we said was that was normal. Just check it the right way around. Which one did I say that the yellow was? Normal eyes and black body. Yeah. Black body. And the green was white eye and normal body. So there we get something different. We would expect them to be, you get the 3 of them to 1 ratio to 0 to 0, and you wouldn't get any of those. Okay. So that would show that those two genes are on the same chromosome. Now, so there's two possibilities really. We can either get, there's two extremes. We can get a 9 to 3 to 3 to 1 ratio, or we can get a 3 to none to none to 1 ratio. That's the possibilities. Okay, if I double that up, let's just multiply everything by 9. So that's the same as having, just to make it a bit easier, that's the same as having a 9 to 0 to 0 to 3 ratio. Okay, so we're either going to get this or that. But you may know, in actual fact, it's very rare that you get completely that. But what is possible is very often you see is you get a 9 to, I'm just going to put the numbers in, a 9 to 1 to 1 to 3 ratio. So you do get these two, but they're much rarer. Now, how could that happen? The way that happens is if these two genes, and I'm going to go back to this diagram here again now. Okay. Oh, well, sorry, let's go back up. Go back up to this diagram. If these two genes are on the same chromosome, but they're a long way away from each other, you may remember in meiosis, you get this thing of genetic crossover occurring, where they swap bits of chromosome. Now, if that occurs, then you can get these other combinations of gametes, which we said were impossible. And so if the genes are a long way away, then yeah, you can get these other ones which we wouldn't expect. You can get them occurring, but usually you don't get them occurring in such big numbers. You don't get 9 to 3 to 3 to 1. You're going to get 9 to 1 to 1 to 3. And this here, what's happened is the genes, we've had some crossover, and that means the genes are on the same chromosome, but they're quite close to each other. They're quite close on the chromosome, so crossover, sorry, they're not quite close at all. They're a long way away from each other, beg your pardon. So because they're separated, so crossover is possible. With this one, if you get that, they must be on separate chromosomes. And if you get this extreme where you don't get any of these, you know, those two, then you say that they're on the same chromosome, but they're extremely close to each other. The genes are very close to each other on the chromosome. And the bit of jargon for that is we say that there is strong linkage. Okay, so they don't tend to get separated in meiosis because they're too close, so the chance of it crossing over would be really slim. So I'll just draw a little diagram to explain that. Right, here is two chromosomes, homologous pair. Let's say you've got two genes, you've got that blue one and the blue one, and you've got a green one really close to it. Now, if they're to swap bits of chromosome, the crossover would actually have to happen there, just on one tiny bit of the chromosome, and that's not very likely. Whereas let's draw the same thing occurring here if you had them separated by the genes are separated by a bigger distance. So I'll draw the blue one there and the green one there. Let's say that we get a crossover event, and it can happen anywhere in that band there. It can happen anywhere at all there, so it's much more likely. Let's say that it just happens here, draw a little crossover. So what we're going to get is we will get red. Okay, so what's happened? They've swapped over bits of genetic information there, and so you could end up with the two genes which were from the same chromosome being separated, and that's quite likely because they say any crossover in that region there will cause that. Whereas this one, you'd have to have a crossover in a tiny little region, which is much less likely. So you tend not to see that happening.